Regulation of gene expression refers to controlling the timing of appearance and amount of protein that is synthesized. Gene expression results in the formation of a polypeptide, which is why it can be regulated at several levels. For instance, the different colored patches on a calico cat are due to different levels of expression of pigmentation genes in various parts of the skin. In cats and other eukaryotes, genetic regulation occurs at the following levels: transcription, post-transcriptional modification, transport of the mRNA from nucleus to cytoplasm, and translation. While in prokaryotes such as E. coli, the predominant site of gene regulation is the initiation of transcription. Therefore, we see that gene expression is regulated differently in eukaryotes and prokaryotes. Since gene expression results in the formation of a protein, genetic regulation controls which protein must be synthesized and which protein need not. For example, E. coli can use glucose, a monosaccharide, or lactose, a disaccharide, as its source of energy. However, lactose needs to be hydrolyzed or digested before it is used, and hence the bacterium prefers to use glucose when it can. But in the absence of glucose and presence of lactose, E. coli synthesizes an enzyme, beta-galactosidase, to catalyze the hydrolysis of lactose into galactose and glucose. However, in the absence of lactose, the bacteria will not synthesize the enzyme beta-galactosidase. This shows that gene expression depends on the metabolic, physiological, or environmental conditions that exist in the cell. Moreover, gene regulation allows the cell to control its structure and function. This is the basis of cellular differentiation, morphogenesis, and adaptability of any organism. In fact, the development and differentiation of an embryo into an adult is also the result of coordinated regulation of expression in several sets of genes. In prokaryotes, the predominant site of gene regulation is the initiation of transcription. In the transcription unit, the interaction with accessory or regulatory proteins regulates the ability of RNA polymerase to recognize and bind to a promoter, that is, the start sites. When regulatory proteins act positively, they are called activators. And when they act negatively, they are called repressors. The accessibility of promoter regions on the DNA in many cases is regulated by the interaction of proteins with sequences called operators. The operator region lies adjacent to the promoter. In most cases, a repressor protein binds to the operator sequence, due to which the operator is inactivated and hence transcription does not take place. But when the repressor protein is removed, it activates the operator and hence transcription occurs. The regulation of gene expression can be studied by the operon hypothesis. Let us now understand genetic regulation with the example of lactose or lac operon in E. coli. The lac operon was described in detail by two people, a geneticist, François Jacob, and a biochemist, Jacques. They were also the first to explain a transcriptionally regulated system. The bacterium E. coli uses either glucose or lactose as a source of energy. Depending on the source of energy available, there are 4 possible situations that exist in the bacterium, as shown in the table. Before we understand the lac operon in detail, let us understand what an operon is. An operon consists of a structural gene which codes for several proteins and is regulated by a common promoter and regulatory genes. Each operon has its specific operator and specific repressor. For example, the lac operator is present only in the lac operon and it interacts specifically with the lac repressor. The lac operon consists of 1 regulatory gene, or I gene, and 3 structural genes, lac Z, lac Y, and lac A. The I gene codes for the repressor. The structural genes code for enzymes required for the metabolism of lactose. The lac Z gene codes for beta-galactosidase, which hydrolyzes lactose into galactose and glucose. The lac Y gene codes for permease, which increases the permeability of the cell to lactose, while the lac A gene codes for trans-acetylase, whose function is unknown. The switching on and off of the operon is controlled by lactose and is hence the inducer. Since lactose needs a small amount of permease enzyme to enter the cell, a very low-level expression of the lac operon is continuously required. The lac operon functions in the following manner. The repressor protein is continuously synthesized from the I gene. The repressor in its active state binds to the operator sequence, thus preventing RNA polymerase from binding to the promoter site and transcribing the operon. In the presence of the inducer lactose or allolactose, the repressor is inactivated and transcription proceeds. Thus, regulation of the lac operon is an example of regulation of enzyme synthesis by its substrate, that is, synthesis of the enzyme beta-galactosidase by its substrate lactose. This regulation of the lac operon by the repressor is called negative regulation. In this way, the lac operon demonstrates how gene expression is regulated in an organism.